Well, thank you for that, and thank you to the conference organizers as well. I've really enjoyed myself here, and excited to give this talk. So as what was alluded to, this is going to be a joint conversation right here. Like a lot of science these days, there's a lot of collaborators, right? And so my friend and colleague, Mark Ebert, is one of those collaborators on this project. So I'll give a brief overview of what we did, and then he'll get into kind of the meat and potatoes, so to speak, on the algorithms and the results and everything. We don't have any relevant disclosures here, but the session organizers did ask us to talk about some of the lessons that we've learned collaborating with non-computational biologists over the years. So just broadly, I think that it's so critically important to be able to speak the language of biology, right? That's the domain-specific biology of whatever sort of computational analyses you're doing. You need to be able to understand what they mean, because if you don't, then the value of your research is just going to be so limited, right? And what I really mean by that is be slow and methodical in your study design and in your results and in your presentation of everything, because something that makes sense to me as a computational biologist might not make sense to the pure biologists that we're collaborating with, and vice versa, right? Things that might be really obvious to them might not be intuitive to me. So let's get into kind of the background for our research. Over some of the other sessions on the other days, some of these nucleotide transformer models have been talked about a little bit. And what are these foundation models? Well, in a nutshell, a DNA nucleotide transformer model is trying to represent what normal DNA looks like. And so then we can create some sort of embedding that can be used to predict some sort of biological feature. Now, the figure that we have up here comes from the nucleotide transformer, which was not from our lab. That was from a different lab. But the nucleotide transformer is very similar to some of these other foundation models, whether it's hyena DNA or DNA-BERT or Grover or any of these other foundation models. They take in a sequence of nucleotides, and then they tokenize them, right? And that tokenization step for the nucleotide transformer is you take 6 nucleotides and you condense it down into a single token. And then you repeat for the next 6 nucleotides, which has a token. All of those tokens are then passed into the trained model, okay? So we have a pre-trained and a frozen model, and then that creates these embeddings. The embeddings can then be passed into the prediction head, which can predict out whether something's an exon or an intron or promoter or some sort of biological signal from the DNA. Now, SegmentNT in particular is important for our analyses. SegmentNT takes that one step further and asks, can we predict at a nucleotide resolution the inclusion or the exclusion into one of these different features? So is this specific nucleotide belonging to an exon or an intron? And the way it does that is through rope embeddings and a UNet architecture, which I display up here. Again, this is straight from the nucleotide transformer and SegmentNT papers. They're from the same group. They're all included in the same GitHub repository, right? What's important here is that as opposed to the traditional autoencoder, the UNet has these skip connections between previous layers and the output layer to be able to have absolute positioning in addition to the relative positions of the lower layers. So why do we care? Okay, I think that this is so important right here. Like, why do we care about nucleotide transformers, SegmentNT, all these transformer models and everything? It's because there's a lot about DNA that we don't understand. There's a lot about this basic biology of these DNA sequences that we have no idea what they're doing, right? Why do we have these DNA sequences? Well, what are these nucleotides really doing? right? And when it comes to individual risk prediction, we know even less. When the average individual out there has 5 million single nucleotide polymorphisms, right? And most of those don't really have clear annotations from previous studies. Like, we don't know how these are interacting together. We don't know what the disease relevance might be for all of these combinations of variants. And that's something that these transformer models can really get to. At its crux, the nucleotide transformer and SegmentNT can find these combinations of variants that might be disease relevant. But in order to get there, we need to make sure that the predictions are accurate. We need to make sure that they're consistent, that they get the exact same results every single time. And that's what our study here is going to address, and that's what Mark is going to be talking about in terms of our results, right? We asked ourselves, so are nucleotide level intron-exon predictions accurate? Are they consistent? And at what point is the context window sufficiently large to accurately predict nucleotide level annotations? And is this sensitive to the change in the context? How sensitive is that? If you just add in an extra token or two here or there, is that prediction going to be changing, or is it not? And finally, we also looked at other biases that exist in these models currently and how we might be able to overcome those. So with that, I'll turn the time over to Mark to get into our results.
Wonderful, thank you. See if I can get the laser pointer here. Okay, so our study had, the basic purpose of our study was to assess underlying biases in SegmentNT, specifically to input sequence length and the position of a given nucleotide within that input sequence. And we did this by selecting 6 genomic regions, including 5 genes and 1 negative non-genic control. Here we're showing the gene structures for the 5 genes based on NCBI RefSeq annotations. The pink boxes are the exons and the black lines are introns. And we're also indicating the location of a specific nucleotide that will be used in one of our analyses for each gene. Presentation will be focusing on the results from the ApoE gene. So the 2 basic analyses that we performed are, the first was looking at, for every nucleotide in these genes, how did the probabilities of being either intronic or exonic change based on whether that nucleotide, that same nucleotide, was in the first position, the middle position, or the last position of that input sequence. And second, this is a more in-depth, so the first one is all the nucleotides in a gene. The second one is going to be looking at a specific nucleotide and how its probabilities change based on being in every position in the input sequence. So first, second, and so on until the last. So from this data, we're able to perform several formalized analyses to assess the biases of input sequence length and position within the sequence, including model performance. So let's get into the data. The first we're going to look at is nucleotide position. So whether that nucleotide is first, middle, or last, and how it biases SegmentNT probabilities. And what we're going to show is that each position results in a very distinct probability distribution that requires a unique interpretation. So what we're looking at here is ApoE. Along the x-axis is the nucleotide position within ApoE, so all the nucleotides in ApoE. And ApoE is a top Alzheimer's disease gene. If you're not familiar with it, that's an area of research for us. And on the y-axis is the probability of each nucleotide being in an exon. I think the mouse froze. There we go. And along the top is the gene structure based on RefSeq, and it's aligned appropriate with the nucleotide positions. So what we're showing here is that whether the nucleotide is first, which is purple distribution, middle, red, or last, green, you get a completely different probability distribution. So just to make that clear, this is at one nucleotide. In the middle, you got a probability of like close to 1. If it's in the last position, it's like 0.1. Same nucleotide, right? So we also formalized this statistically just to demonstrate that each of these is a unique distribution or distinct distribution, whether it's first, middle, or last. So the first conclusion that we're showing here is that a single threshold cannot be reliably set for raw SegmentNT probabilities. So we then wanted to know, would it be possible to normalize these so that you can set a specific threshold, so you can interpret them uniformly? And the short answer is yes. We did two simplistic approaches. The first is we took the z-scores of all these probabilities. We're showing here is that this immediately puts them all three on the same scale. You can look at them, you see the same basic trends. And setting a single threshold of z-score of 0 for all of them, we can then test model accuracy for each, whether the nucleotide is in each position. They all perform quite well, greater than 90% accuracy. Middle performs best with approximately near 99% accuracy. And then looking at the AUCs, the ROC curves and the AUCs associated with those, again, all three perform quite well, with the middle performing the best with 0.999 area under the curve. The next we looked at was another fairly simplistic approach of just subtracting exon probability minus the intron probability. And in this case, to the naked eye, there's still very distinct distributions, especially when you look at the last position. It looks, very compressed. It doesn't look that great. But again, if we set a single threshold for all three of them at 0, they all three perform even better than the z-score approach. Again, where middle performs best at 99%. Again, with the AUCs, they all perform quite well. Again, middle performing best. So the second conclusion is that SegmentNT probabilities can be interpreted more uniformly when normalized, but the middle still performs best. So moving on, to this point, we were looking at all these nucleotides, whether it's first, middle, or last. Now we're going to look at a single nucleotide, moving it progressively through every position in the input sequence, and see how these probabilities fluctuate for a single nucleotide. Got to make sure that is clear. And there are two main things that we're going to point out here. But first, what are we looking at? We're again looking at ApoE. Up here is the probability of being an exon, and on the same plot, the probability of being an intron. Along the x-axis is the position in the window, not in ApoE, but a single nucleotide at every position of a 24 kb window. Along the y-axis is, again, the probabilities. So the two things that we want to point out is if you look at these probabilities, if everything was working perfectly, we should get the same probability throughout this whole plot, but it is not. You look at the thickness of this band of probabilities and this one of intron, it's bouncing all over the place. I mean, it looks like one blob of data because there's just so much bouncing around. That's the first thing. So it's bouncing. Second is that towards the beginning and the end of where it is in the window, it's less stable. So the probability for being an exon drops dramatically towards the beginning and towards the end. And it's just really sporadic for both of them on both ends. So here we're going to zoom in and see exactly what this looks like. And the first thing that really stands out to you is that, or at least to us, was that it just seems to be oscillating. And on an apparent 6 nucleotide cycle, just up and down, following the exact same pattern, it seems. Second, what's really astounding to us is that by shifting the same nucleotide only 2 positions, you get a 4% difference in its probability of being an exon. 2 positions, same nucleotide. For introns, shifting it 4 positions, you get greater than a 20% difference in its probability. So it's really, it's bouncing, right? So as I mentioned, initially it looks like as though this is a 6 nucleotide repeat cycle, but we're going to show it actually is a 24 nucleotide repeat cycle. So taking the same data that we were just looking at, rather than plotting all these plots on a single line, we're going to now break that up into 6 lines, plotting every 6th value. So if we do that, if this is really a 6 nucleotide repeat cycle, if we break that into 6 lines, plotting every 6th value, we should get 6 flat lines. But that's not what happens. There still remains a 4 token repeat cycle. So from there, we progressed to a 24. Instead of plotting every 6th value, we're going to plot every 24th value. And here you'll see that we get 24 different lines, all of which are completely flat, regardless of whether we're predicting intron or exonic. So really, this is pretty fascinating question. Why is there this oscillation in segment NT probabilities? But the conclusion number 3 is that segment NT probabilities vary widely across only a few positional changes for the same nucleotide, especially towards the ends, and they oscillate, i.e. raw values are unreliable. So the last thing we want to look at is how does input sequence length bias segment NT probabilities? This is something that I think we would expect to be a fairly critical component, and indeed it is, though we were a bit surprised by the results. So starting with only a 24 nucleotide input sequence, again, this is ApoE, just looking at the probabilities of being an exon. It just looks pretty sporadic. None of these spikes are correlating with the introns and exons of ApoE. Moving up to a 192 nucleotide input sequence, it seems maybe less random, but still not a great correlation between introns and exons. No peaks for exons or valleys for introns. Going from 192 up to 768, now we're starting to see some separation, a pretty clear separation. It's a sudden spike. And I should emphasize, we looked at 11 different input sequence sizes. Here I'm just showing 4 representative, but I'll show you the data, kind of a summary data for all 11 here in a minute. And then going up to our max input sequence size of 24 kb, there's very clear peaks and valleys with exons and introns respectively, as we saw earlier. So the question is, at what point do we maximize value from the input sequence? Does it always get better no matter how big the sequence gets, the input sequence gets? The answer is no. There is a point where it quickly we maximize the utility. So here along the x-axis, we're looking at the 11 different input sequence sizes that we included. And along the y-axis is the model AUC based on the raw middle exon prediction value. And each line represents one of the 5 genes. And in the beginning, there's quite a bit of variability in terms of how well the model performs. Looking at the point where each gene surpasses 0.9 AUC, it's actually at a different point. So for ApoE, you only needed 384 input sequence. For TNF, it was 768, and for the other 3, 1536. So there's variability across genes, which is also a fascinating question. But ultimately, all 5 genes quickly plateau at only 3,072 nucleotide input sequence lengths. And so going beyond that, there's very little, if any, benefit, which has important implications for both training and usage. So conclusion number 4, input length does matter, but we were surprised at the point at which it plateaued. So before I just summarize our conclusions, I just wanted to thank everyone who contributed to this, in particular Anna Ho, who laid the foundation for this work. She's a graduate student in Dr. Miller's lab, along with our other collaborators and lab members. And then just to summarize, I'll just put these up here, our 4 conclusions, and we'll be happy to take any questions.
Get started. Great talk. Thank you, both of you. I have many questions I could ask, but I suppose the one I'll start with is thinking about the nature of foundation modeling, right? Because you were talking about the utility of a lot of these transformers models as we move towards foundational models for biology. But I also think here you actually produce a pretty probabilistic stance that could be used in a sort of antithetically to foundation model way, which is like we have priors for things, right? Which I think people are trying to get away from. So I just was hoping from your perspective as someone who both sees perhaps the direction of foundation modeling for biology, but somebody who's also coming back and investigating things from a more probabilistic standpoint, how do you reconcile these things?
That's a good question. Do you want to take that one, Justin? Yeah. Let's take that one.
Yeah, no, I think that's a really good question. I think that I think that's well known that there are issues with with large language models, and those things have been pretty well documented in terms of hallucinations, in terms of consistency and everything. And you're absolutely right that the field is moving away from probabilistic approaches. Yet, if we are going to apply this to the clinic, clinicians still want that probabilistic value, right? They still want to be certain that the results are accurate. And so we have to do some sort of formal analysis to show that these results are useful. And that's really the crux of what we're getting at here is, are there any biases in these models right now? And if there are there any ways that we might be able to improve the approaches in the future?
So, yeah, great presentation. So do you think how the model was trained might be the cause for this like variation in terms of like the position of the token here? And maybe like data augmentation somehow in the way that it's fed similar to the CNNs, like adding noise or rotation or transformation, like whatever it's done in images can improve the overall performance.
Yeah, no, that's a great question right there. And we've hypothesized several different reasons for these oscillation biases in particular that we're seeing. And I think that you kind of hit the nail on the head that the way that these models are being trained right now is fundamentally producing these biases that we're observing. And obviously, we just looked at the nucleotide transformer and SegmentNT here, but a lot of the foundation models are kind of built on the same premise, right, of this tokenization approach that then gets passed into some sort of UNet or rope embedding approach to then predict things afterwards. And yes, here we're seeing a token length of 6 nucleotides, right? And as Mark was showing, it looks like the oscillation occurs every 6 nucleotides. It actually turns out to be 24. In this case, the minimum number of nucleotides for SegmentNT is actually 6 times 2 raised to the power of n. So every context length needs to be of that length, which 2 raised to the power of 2 for 4 times 6 equals the 24. So we hypothesize that that's the reason that we're seeing the 24 nucleotide oscillations because of that training. But again, many of these foundation models do something really similar. They might change the length of the tokens. They might change some of the requirements in terms of, is it 2 raised to the power of n, or is it 4 raised to the power of n, or whatever it is, right? But we need to evaluate any of these models to see, are these predictions consistent, or are they oscillating like what we're showing right here?